Hello, we would like to welcome you to this webinar investigating a complex polymorph and pseudo-polymorph landscape using ReactRaman. We are
Andreas Stromberger
and Christian Herz, and we are technicians from Excella. At first, we would like to give you a short overview of this webinar's content. First, about Fareva and Excella. Second, the polymorphism, the challenges. Third, the starting point with the current process. Fourth, overcome of limited process understanding with the use of PET. And finally, the challenges resolved with ReactRaman.
giving you a short background to our company and the Fareva holding. Fareva is one of the world's leading subcontractors with 39 production sites in the cosmetics, pharmaceuticals, makeup and industrial and home care fields. We are a family-owned enterprise whose strength lies in its independence, and we are a community of women and men guided by a commitment to excellence. At your service. Excella is part of the Fareva Group since 2008. We are located in Feucht, which is in Germany and is 20 kilometers southeast of Nuremberg and 160 kilometers northwest of Munich. Excella was founded in 1913 of the pastor Ludwig Heumann. We were also a family-owned company till 1990. Then we were part of several big pharma companies like Searle, Pharmacia and also Pfizer. Since 2008, we are part of Fareva. Excella supports two business units, drug product and drug substance, or the so-called API business. We are fully approved and certified like by EMEA and FDA and others.
Excella disposes of a lot of core competencies to offer a one-stop shop concept for the customer. The drug substance API division offers 40 years of experience with over 80 molecules from different structural classes and is capable to handle critical actives. The drug product pharma division of Excella provides contract manufacturing services, especially for the development, clinical supply and commercial manufacturing of solid dosage forms containing cytotoxics, high potent or other critical actives with low occupational exposure levels. Please be invited to confirm our unique profile as experts in one-stop shopping services of high potent products. Working with Excella means immediate and easy availability of both drug substance and drug product. We are able to tackle challenges immediately and in close cooperation. We have a one-stop shop concept with all-inclusive prices. Our core competencies in the drug substance API field are: we can offer smart chemistry for complex and difficult molecules, multi-step synthesis. We have high containment labs for R&D and QC. We also have state-of-the-art production lines for high-potent drug substance with 3 independent compartments for OEB4, 5 and 5+. 250 and 800 liters multi-purpose plant with SafeBridge certification. Large-scale manufacturing 4,000 liters up to OEB4. Milling and micronization of high-potent APIs. Scale range from 1 kilogram to 15 tons. Controlled substances supporting NDA and NDA filings. Please feel free to contact us for a good cooperation. We are a reliable partner.
Now we want to start with the topic of our webinar, investigating a complex polymorph and pseudo-polymorph landscape using ReactRaman. At first, I want to give you a short summary of the following topics. I will describe the API manufacturing process and how we detected various polymorph transformations during crystallization and drying steps. Then how the final API processing often yields undesired polymorphs and pseudo-polymorphs, and how we decided to use crystallization process development equipment of METTLER TOLEDO AutoChem like LabMax, FBRM, ParticleView and ReactIR. The process trends and images showed morphology changes, but the complex polymorphic landscape remained difficult to understand and control, so we finally decided to evaluate the new ReactRaman probe. The original process was developed in 2003 and had no control on PSD, delivering relatively big particles. A D90 above 300 microns. As you can see, there is also a bimodal distribution in this picture of the master sizer particle size distribution. Of course, no PUD was used in 2003. Due to customer request in 2018, the crystallization process had to be optimized in order to achieve a control small particle size distribution to avoid the additional milling step. The customer wants to have a D90 below 50 microns out of the crystallization process. But during the optimization and a running production campaign, we also noticed that the desired polymorph is the first thing to control, which wasn't a topic in 2003.
This is the process description of 2003. The API is dissolved in 2 volumes of ethanol at 45 degrees centigrade. Then a Polish filtration using charcoal as a filter aid is done. Afterwards, cooling to 35 degrees centigrade to get supersaturation. Then spontaneous crystallization occurred or optional seeding was done. Then additional stirring time at 30 to 35 degrees centigrade for 30 minutes. Afterwards, cooling to 0 degrees centigrade and additional stirring time for 1 hour. Then filtration and washing, drying at 60 degrees centigrade, and finally in-process control of water with a maximum of 0.5%. After the first production in 2003, 15 years passed till we started again to produce the API in 2018. The batches of the 2018 campaign showed borderline or even OOS results with regard to the residual solvent ethanol. The IR identity analysis was also conspicuous but still met the specification. Offline XRPD analysis gave evidence of a new polymorphic form of the API. At that point, it was obvious that not only the PSD had to be optimized, but much more the desired polymorphic form.
First experiments in the lab to get a finer PSD, which were close to the start of the production campaign in 2018, showed also some strange peculiarities like water content, IR identity, and the melting point. Different seeding protocols did not seem to have an influence on the final PSD. Although we used different seed qualities, seed amounts, and seeding procedures like wet seeding. The idea of alternative solvent system resulted in other difficult issues. All in all, the experiments were not reproducible. To make matters worse, all those experiments yielded different polymorphic forms. The conclusion was to get a deeper process understanding to control the polymorphic form and the BSD. At that point, it was totally obvious to work with crystallization workstation and PUD. So what was the output of PUD? Here you can see the process investigated with PUD like FBRM, ReactIR and PVM. Let me give you some information at first. The red line is the temperature of the reactor. The brownish line represents the corresponding IR peak of the API. The yellow and blue line shows the FBRM trends of the fine and bigger particles. Let me explain the process. The API was dissolved at reflux and was then cooled to 35 degrees centigrade. Heating at 35 degrees centigrade initiates crystallization. We can see the FBRM trend increase, the IR trend drops or decreases. The PVM shows relative small particles at point 1. While 1 hour hold time, particle growth and reduction in fines can be recognized. Then a cooling ramp is started. Starting at 90 degrees centigrade, an unexpected process change can be observed with an exothermic behavior. The FBRM trend shows an repeatedly increase of the bigger particles and a drop of the fines. The IR trend is also dropping. Big particles are noticeable in the particle view pictures at point 2. At the third point, which is after finishing the cooling ramp, the process change is finished. This is an indication of polymorph transformation. Taking a deeper look in the transformation event at the time point 3 hours 10 minutes. This unexpected exotherm event changed the particle population dramatically. There is also a change in the particle morphology and a sudden drop in the solution concentration. All in all, this is a strong indicator for polymorph transformation. But representative sampling is very difficult due to downstream issues like drying and afterwards offline analysis. After having several consultations with METTLER TOLEDO AutoChem, it was decided to test the new Raman probe in order to get a deeper understanding of the transformation and the process.
We began our experiment performing Raman spectra both for the desired and undesired form of the API. This is one of the big advantages of the Raman probe, to measure offline all existing polymorphs very easy and very fast. This means simply measuring the solids before starting the experiment. This overlay shows us the desired form in green and the undesired form in yellow. With this information, we were able to track the transformation event by ReactRaman online. This picture shows the Raman results. Here, Raman peaks were trended over time, with the desired form shown in green and the non-desired form in orange. Initially, the crystal form is shown to be a one-to-one mixture of both polymorphs and are fully dissolved. Most likely, the desired form is even transforming into the non-desired form while heating and start of the dissolution process. Following the same temperature ramp as in the figure in slide 12, crystallization is again initiated through seeding at 35 degrees centigrade. Interestingly, the undesired form can be seen to crystallize first, indicating that this is the least stable form as proposed by Ostwald's rule. Then, an absolutely new information can be obtained with a new Raman trend. It can be seen the appearance of an intermediate form far ahead the exothermic event. But this intermediate is disappearing right at the starting of the exothermic event at 19 degrees centigrade. At that point, the Raman probe identifies the start of the formation of the desired polymorphic form. This new process knowledge can just be obtained by using the Raman probe. Just working with FBRM, PVM and IR doesn't describe you the existence of the intermediate form. In the meantime, we could clarify that this intermediate form is a pseudo-polymorph, an ethanolate. Raman revealed that the crystal form undergoes a transition via an ethanolate and that this ethanolate, a solvomorphism phenomenon, drives the formation of the desired crystal morphology. In-situ Raman was used to elucidate the formation of the desired polymorphic form. ReactRaman revealed that the desired form appears via an ethanolate intermediate which wasn't known before and would be difficult to detect via offline techniques. ReactRaman increased reaction understanding through advanced in-situ analysis capabilities and is especially valuable when liquid and solid phase analysis are of interest. After gathering this process knowledge, we were able to produce the desired polymorphic form. But achieving the desired small PSD is still work in progress. The experimental work with the ReactRaman gave us a clear process guideline how to manufacture the desired polymorph in our next production campaign. This brings us to the end of our webinar. We hope that the presentation was worthwhile and interesting. So we would like to thank METTLER TOLEDO, Dr. Schacht, Dr. Schoss, Dr. Kasakov and Dr. Jaurich.
And Excella, Dr. Nössner, Dr. Jagusch, Dr. Hoffmann and Mrs. Sinden. Thank you very much and goodbye.